All right, ladies and gentlemen, I'm back again, whether you like it or not. One of the things that you're going to have to be able to do is transcription and translation. Now, in this question, this is a release question. They give you the mRNA codon chart, but they also give you mRNA down here. I don't know why my thing is doing that. So they're giving you mRNA in both places. That makes things much simpler. All you have to do is translate. So you're going to take the mRNA code CAG and figure out what protein or what amino acid that says. This should say amino acid, not protein. Okay. So is CAG. So you're going to start here. So that first C tells you it has to be in this section somewhere. Okay. Let me use, oops. Oh. All right. That did not do as expected. So I'm going to try the highlighter instead. All right. So there we go. So it has to be in here somewhere. Okay. And then the second letter, the A, tells you it has to be in here somewhere. Okay. So now we know it has to be in this block. And this third letter, the G, tells us that it has to be in this row. So it has to be right there. It has to be that glutamine. Okay. But that's what you're doing. Okay. And now, if they had given you, like, let's say the DNA sequence was ACT. Well, then you've got to transcribe that to mRNA. So A is going to bond to U in RNA, C will bond to G, and T will bond to A. All right, so now our mRNA is UGA. So we're going to look at our chart. Here's the first one, the first U. Here's the second U. Here's the third U. So where do all three of those meet? Okay. Oh, I'm sorry. I did that wrong. All right. It's UGA. So there's the U. Here's the G. Here's the A. So the only place those all show up is right here. And that's a stop codon. So that means that the enzymes reading the DNA stop reading the DNA or copying the DNA or whatever. Okay. So that just happened to be a stop codon. All right. I didn't intend for that, but it did. All right. Let's say let's change that and let's say this was G. All right. And so now the G would bond to a C. So we're going to put a C here. All right. And so now let's look for UGC. So here's U and G and C. So that codes for cysteine. Okay. And that's how you would read it. Okay. So then the amino acid would be cysteine or cysteine. All right. So that's how you read those charts. And they will be on the EOC. I guarantee it. You'll also see something like this. So this is gel electrophoresis. So what they do is they take a gel, a square of gel, like gelatin, but not edible. All right. And then there's these wells at one end of it. And each well gets a little bit of DNA in there. And then they run an electric current through the DNA. Okay. And that electric current causes the DNA molecules or fragments of DNA to move down the gel. Now, the larger molecules are not going to move very far, and the small molecules will move the farthest. So we know that these are like larger molecules up here, and these are smaller molecules down here. That's how they produce these images, okay? And this is how they do paternity tests and crime scene DNA analysis and different things like that. In this case, a hospital wants to identify the parents of a baby based on DNA fingerprints. So, you've got the mom and you've got the dad, and half of mom's DNA goes into the baby, and half of dad's DNA goes into the baby, right? So we need to find the couple whose DNA best matches the baby's DNA, all right? So if we look at this first block here, okay, it matches with 4, and it matches with 5, and it matches with 7 and 8. All right, this block here matches with 3 and with 6. This matches with 2 and 3 and 6. This one matches with 2, and this one matches with 1 and 3, and I think that's it. All right, so the couple that is most likely to be the parents of the baby is couple X. There's a match here, there's a match here, there's a match here, 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 and here. Okay, so that's how you would look for this. You're looking for the suspect or the couple or the organism that is most similar to the evidence. Okay. Now, moving on, kind of quickly, going to run through some things. We did that. Again, one way they might ask you about this, instead of having a baby and parents, they might have a newly discovered organism and other similar organisms. Like, they find some kind of new zebra. So then they give you zebra and horse and donkey. And you have to figure out which one this new type of zebra is most similar to. Okay. The more similar the DNA, the more closely related they are. All right. So let me say that again for the people in the back. Oops. More similar the DNA. Okay. The more similar the organisms. Okay. If we want to know how similar organisms are, we're going to look at the DNA. All right. Now, we've talked about genotype, oops, and phenotype. And I remember that the genotype are the actual genes. And the phenotype, which starts with PH, is the physical trait. So, for example, here you might be heterozygous for brown hair, right? So maybe the big B is brown and the little b is blonde. I am just making this up. I don't know if blonde is recessive or not. But let's say it is for the sake of review. So you might be heterozygous for brown and for blonde, but your physical trait is going to be brown hair because that dominant trait covers up the blonde trait. Okay. If you were homozygous dominant, then you would also have brown hair because that's the only genes you have. If you had if you were homozygous recessive, then you would have blonde hair because that's how that works. Okay. Let's see here. All right. Now, The genotype is what you're born with. The phenotype can be changed or affected by the environment. For example, poor nutrition poor nutrition will not change your genotype, but poor nutrition can change your phenotype. You might have the genotype for the best lungs on earth, but if you spent your life working in a coal mine, your lungs are probably going to be damaged. You may have the genotype to have very low risk of cancer, but if you spend your life smoking, you're increasing that chance of cancer because your environment, the smoking, is changing your phenotype. Okay. All right. And then we talked about Punnett squares. Okay. So Punnett squares look something like this. You have one person up here, or one organism up there, and you have another organism here. And all you do is you take this guy and you bring it over, and you take this allele and you bring it over, and you take this allele up here and bring it down, and you take this allele up here and you bring it down. Now, what this tells you is there are 4 possible outcomes, right? When you mate and you have independent assortment. So these are the 4 outcomes. Now it happens that these 2 are the same and these 2 are the same. So you have your genotype is 50% homozygous dominant and 50% heterozygous. Now your phenotype, and because this is 2 out of 4 and this is 2 out of 4, so it's 50%. The phenotype, okay, you have 2 out of 4 or 50% that are going to have brown hair, and then you have 2 out of 4. Actually, well, you have 2 out of 4. These 2 will have brown hair, but these will also have brown hair. So really, you have 100% brown hair. You have 4 out of 4 that will have the phenotype, the physical characteristic of brown hair. Okay. Okay. And then we can talk about the genotype ratio. You have a 2 to 2 ratio, or you might even say a 1 to 1 ratio of homozygous. Well, let me rewrite that. Where's the eraser? You have a 1 homozygous dominant for every 1 heterozygous. All right, so that's a 1 to 1 ratio. Down here, you have a 4 brown hair to 0 blonde hair people. Okay, now again, this does not mean that if you have one child, you can guarantee with certainty what genotype that child will be. What it means is that if you have a child, whether it's your first child or your 10th child, there's a 50% chance they will have a homozygous dominant genotype or a 50% chance they have a heterozygous phenotype. Okay. Now, if you had 1,000 children, you'd be very tired. But if you had 1,000 children, then you would probably have about 500 that were this and about 500 that were this. But we can't do that as humans. We don't live long enough. We've talked about mitosis and meiosis. Remember that mitosis is 1 division, meiosis is 2 divisions. Independent assortment. So you have a cell that's So we're doing meiosis right now. And we have one cell and it divides, and then those two cells divide again, and we get 4 cells. Now, this cell is going to have 2 chromosomes in it. Let's say A and B. And these will also have A and B in it. And this will have A and B. Actually, this will have 2 copies of A and B. So they separate, and now we have A and B in this cell and A and B in this cell. These will have, this one will have A, this one will have B, this will have A, and this will have B. So this is the independent assortment. Okay. And all it means is that the genes on the A are not dependent on the genes on B. So they can independently assort. They can separate and go to different gametes. These would be the gametes down here. Okay. So that's independent assortment. All right. And then we talked about sex-linked traits. So a sex-linked trait is on the sex chromosomes. Remember, females, biologically speaking, I'm not trying to offend or say anyone is or isn't something. I'm just saying, in terms of biology, a male is XX, okay? And I'm sorry, a female is XX, and a male is XY, okay? Now, so if a gene is on the X chromosome, we say it's sex-linked. For example, normal vision, let's say that is a big C, normal color vision. And let's say a little c is colorblind. Okay. Now, if a female, so we could have a female that is homozygous dominant for normal color vision. Or we could have a female that is homozygous recessive. Okay, so this is normal vision. This is colorblind. All right. And we could have a female that is heterozygous. They will have normal vision. Okay. So, but a male only has one X chromosome. So they have the normal gene for normal color vision. They will have normal vision, normal color vision. If the male has a recessive gene for color blindness, they will be colorblind. End of story. A male cannot be heterozygous or colorblindness because they only have one X chromosome. Okay, that's what it means to be sex-linked. One sex, XX or XY, has a higher chance of having the trait because it's sex-linked. Okay, and then we also talked about codominance and incomplete dominance. So codominance, okay, is where they both show up. So maybe you have a flower, there's a badly drawn flower, all right, and you have another flower, and let's say this flower is blue. I really don't know why it's doing that. And let's say this flower is yellow, okay? And they mate and they produce seeds, and those seeds become a flower, okay? And that flower is green, okay? So what that means is that, nope, I'm sorry, that's not right. I goofed up. Let me undo that, okay? So what would happen is this flower might have some green patches on it, right? And then it might also have some blue patches on it, right? So that's codominance. The colors, both colors show up in the offspring, right? Neither one dominates over the other. They are dominant together, okay? Now, we can also have incomplete dominance. What just happened? Incomplete dominance. All right. Now, with incomplete dominance, maybe you have, let's go back to that previous scenario. You have a blue flower and you have a yellow flower, and they mate, and the offspring ends up being a green flower. Okay? So that is incomplete dominance because neither is dominant over the other. They both show up together, and you end up with a kind of a blended trait. Okay? So that's incomplete dominance. Trying to move through quickly. I don't know if the screen recorder is recording my little battery warning there or not, but it is. All right. So talking about biotechnology. So let's say we have a gene that we want, like to make human insulin. What we're going to do is we're going to take that DNA, right, and we're going to add a restriction enzyme. Now that restriction enzyme is going to cut the DNA so that all we have is the insulin gene. All right. And then we're going to take a bacteria. Well, that's not. All right. So we're going to take that insulin gene, and then we're also going to take something called a plasmid. Plasmid is a circular piece of DNA. And we're going to we're going to use a restriction enzyme again, and we're going to cut out part of that DNA. Okay, we're going to cut that out, and then we're going to mix these two things together. And what's hopefully will happen is this insulin will take residence in here in the plasmid. Okay, and so now we have a plasmid that contains the gene for human insulin. So now we take that plasmid with our new gene, and we're going to add that to a bacteria. Now it has its own DNA, but after this, it's going to have its DNA and this plasmid with the insulin gene. Okay? And so what happens now is this bacteria cell is going to reproduce, and it's going to make insulin. It doesn't know why, but it's going to make insulin. Okay? And so then all we have to do is isolate the insulin, and we're good to go. All right? And I think that is pretty much everything. I'm going to throw one more word at you, and that's transgenic. Okay? And so transgenic means that you contain a gene from some other organism. So this would be a transgenic bacteria because it contains human DNA. All right? And I believe that is everything that we covered in a nutshell. I'll see you next time.